Welcome to the unit on nucleic acids. Now, nucleic acids form another macromolecule that we find in biology, and the famous nucleic acid that you'll have all heard of will be DNA. To start looking at the structure of nucleic acids, we have to consider that they are constructed of monomers called nucleotides. This word here, nucleotides. And the polymer of the nucleotides is called a polynucleotide. So what we're going to look at first of all is the structure of a nucleotide, and then how those nucleotides are polymerized to form polynucleotides. So dealing with the structure of a nucleotide actually, if I was to look at it chemically, not that you have to know the structure of this, if I was to look at it chemically, a nucleotide consists, or looks like this, consists of three components. One component here is called the phosphate group, and then one component here is called the sugar, okay, which in this case is a pentose sugar, a 5-carbon sugar, and another component there is called a nitrogenous base, of which there are 4, which you'll probably remember from GCSE: adenine A, thymine T, guanine C. And more simplistically, we can represent that as a structure that looks like this, okay, with our pentose sugar here attached to a phosphate group which is attached to the nitrogenous base. Okay, remember there are 4 different types of these bases: A, T, G and C. Okay, so there are 4 different types of nucleotide. Each nucleotide has this part of the nucleotide in common with each other, okay, whereas the bases alter. Okay, so there's 4 different types of nucleotide. Here's another diagram here which shows much the same thing here with our phosphate group, our pentose sugar and our nitrogenous base. Okay. The next thing we're going to look at is how these monomers, nucleotides, how they link together to form the polymer, which is called a polynucleotide. And the answer is very simple. If I find a piece of board here, what happens is that I draw a couple of nucleotides. So here we've got 2 nucleotides, and what happens is that a bond forms between the phosphate and the sugar of the adjacent nucleotide. Okay, and this bond here is called a phosphodiester bond. Okay, and much like all the other bonds that we've come across in macromolecules, it's formed by a condensation reaction, and it's called a phosphodiester bond because it's an ester bond. It's 2 ester bonds, if you like, 1 ester bond here, 1 ester bond there, and it involves a phosphate group, hence phosphodiester bond. Okay. The other thing, which I don't know if is necessarily hugely important, but the bases can be categorized into 2 types of bases. One type is called a purine, okay, and they are structures which have 2 ring structures in them, and I'll show you a diagram of that in a second, whereas the pyrimidines, okay, thymine and cytosine, have 1 ring structures. And if I quickly look at a diagram of that down here, here I've got the purines, which are these double ring structures, one ring, two ring, adenine and guanine, and then we've got the pyrimidines, which are just single ring structures here. Okay, thymine and cytosine. I'll talk about uracil when we talk about RNA in a minute. Now, in terms of base pairing, which we'll talk about in a second, you'll notice that thymine always base pairs with adenine, tennis association, my little mnemonic for that. So you'll notice that a one ring structure always base pairs with a two ring structure, and guanine always base pairs with cytosine. Cytosine always base pairs with guanine, again two ring structure, base pairing with a one ring structure, which means that the distance between the sugar phosphate backbones in our DNA structure is always 3 rings wide. So, summarising all of that, okay, with the idea of nucleic acids, okay, and the fact that they are polymers, which we call polynucleotides, and those polynucleotides are made out of monomers called nucleotides. Okay, we talk about a nucleotide having 3 components: the pentose sugar, the phosphate group, and a base. Okay. And then specifically looking at DNA, for DNA the sugar is called deoxyribose, okay, and the 4 nucleotides involved are adenine, thymine, cytosine and guanine. Of those, 2 of those, adenine and guanine, are a subcategory of bases called purines, whereas thymine and cytosine are a subcategory of bases called pyrimidines. And we also looked to see how these nucleotides polymerize or form polymers by condensation reactions, where a phosphate group binds with a sugar, okay, to form what's called a phosphodiester bond. Moving on to the next slide, this talks about another type of nucleic acid. This one is called RNA. Okay, we'll come back to what the actual function of RNA is, but just for this slide I want to concentrate on the difference in structures between RNA and DNA. And an RNA also consists of a sugar, a phosphate and a base. Okay, but there are differences. If you look at, for a start, the sugar is not deoxyribose as it was in DNA. It consists of a sugar called ribose, and there are 4 bases, but the base thymine doesn't exist. But instead of thymine, we have another base called uracil. Okay, so the bases that we get in RNA are adenine, uracil, cytosine, and guanine. Okay, so uracil replaces thymine, okay, ribose instead of deoxyribose, and RNA is single-stranded molecule rather than double-stranded molecule we get with DNA. RNA tends to be shorter than DNA. Okay, so summarising that with a few diagrams, here I have a polymer of RNA, noticing that the base uracil exists, okay, instead of thymine. That's this pentose sugar here, okay, is ribose rather than deoxyribose, okay, and also notice that the whole structure is single-stranded rather than double-stranded. Okay, another diagram which shows that, a comparison between the two. Here on the right of this diagram I have DNA with its double helical structure, with its bases cytosine, guanine, adenine and thymine, whereas on the left I have my single-stranded RNA molecule with its bases cytosine, guanine, adenine and uracil. Added to that is that the pentose sugar involved in the sugar phosphate backbone is ribose as opposed to deoxyribose in DNA. To the next slide, Okay, this looks at how the structure of a double-stranded molecule of DNA forms. One of the key ideas here is the idea that DNA, the two strands, will bind to each other by what are known as complementary base pairing. Okay, and as I mentioned before, the two bases that we, or the two base pairs that we deal with are adenine, always base pairing with thymine, and guanine, always base pairing with cytosine. Okay, so if you look at this blue ribbon here is representing the sugar phosphate backbone of their DNA, and then stretching into the middle of the DNA molecule we have the nitrogenous bases, which always base pair adenine with thymine or vice versa, and guanine with cytosine or vice versa. The mnemonic I've always used is golf club and tennis association. Okay, the other thing that you should be aware of is that the hydrogen bonds that hold the thymine and adenine, T and A together, there are 2 hydrogen bonds there, okay, where there are 3 hydrogen bonds that hold guanine and cytosine together. Okay, so the little T with the 2 bonds to A and the G with the 3 bonds to C. Okay, the other thing that you should be aware of is the strands run in opposite direction. You'll notice that these pentose sugars here are pointing upwards, whereas the pentose sugars there are pointing downwards. So the strands run in opposite directions, and that feature is known as the fact that they run anti-parallel. Anti-parallel is a word that you should be aware of, anti-parallel. Okay, and then the strands twist. The analogy is often of a ladder with its handrails, vertical handrails, and horizontal rungs. Okay, so you consider that to be a ladder, but the ladder twists to form this helical structure, which is known as a double helix. Double because it's double-stranded, 2 strands, and helix because it forms a twisted coil type structure, or a helix. One of the key features of DNA is that it can form copies of itself, and in fact when you've done the study of mitosis or meiosis, you'll know that before any form of cell division occurs, we need to make a copy of all the genetic material in the cell. That means we have to make a copy of the DNA, and it is a very simple process in which our single piece of DNA is attached to by an enzyme called DNA polymerase. And what the DNA polymerase does is it unzips by breaking the hydrogen bonds between the complementary base pairs. Okay, and it also catalyzes the addition of free nucleotides, so nucleotides, single nucleotides which are present in the nucleus of the cell, come in and they bind to each half of the molecule by complementary base pairing. Okay, and it also catalyzes the phosphodiester bonds in the new strands. Okay, and that results in 2 identical strands being produced. If I look at that or represent that diagrammatically, here's a simple diagram here which is showing a single strand becoming 2 strands. Okay, so the DNA polymerase is here moving up, unwinding, unzipping the single piece of DNA. The free nucleotides are coming in here, base pairing, A to T, G to C, etc., forming 2 strands like that, showing that in a slightly more complicated diagram, in fact possibly too complicated for what you need to know. But here we have our single DNA molecule, and along comes our DNA polymerase. You don't have to know about helicase, which is an enzyme which actually does the unwinding. You don't have to know that enzyme if you just know the enzyme DNA polymerase. So the DNA polymerase comes along, it unzips the strands by separating the hydrogen bonds or breaking the hydrogen bonds, exposing free nucleotides or a free sequence of nucleotides, and in come single nucleotides which base pair to each half, and the result is that we end up with two strands of DNA which are exact replicas of our original strand. The fact that the two new strands of DNA each contain half of the old strand and one completely new strand is a feature known as semi-conservative. Semi meaning half, conservative means that half is conserved in each of the new strands. Okay, if I was to quickly show you a little animation of that happening.
DNA replication is defined as the process by which an organism's original DNA is used as a template for the production of a new complementary DNA strand. An enzyme called helicase unwinds the original DNA's double helix, creating a replication fork. Next, an enzyme named DNA polymerase III works down the leading strand and up the lagging strand of the replication fork, synthesizing two new strands of DNA by taking free nucleotides and pairing them with the complementary bases on the original DNA template. The process of DNA replication is described as semi-conservative because the two copies of DNA produced each contain one strand of the original DNA and one entirely new DNA strand.
Moving on to the next slide here, which looks at the idea of the gene, which we use in biology a lot, and just to orientate yourself into what a gene is within respect to a cell and what it means in terms of its DNA. If you think of our cell here with its nucleus, and as the work you've done in with cell division, you will notice that just before cell division occurs we have these things called chromosomes appear. If I was to look at this as being a chromosome, a gene is a piece of DNA which exists at a specific point on a chromosome, which we call a locus, a gene locus, and that gene is a length of DNA. And what does that DNA do? That DNA encodes for the production of a polypeptide. Okay, so the sequence of bases in the DNA is encoded into a sequence of amino acids in a polypeptide, and it's those polypeptides that fold up into specific shapes and do particular functions in the cell, such as being an enzyme or a carrier protein or an antibody or whatever it is. And it's the functions of those proteins which determines the characteristics of the cells and therefore the characteristic of the organism. And that leads us to an idea which is known as the central dogma of molecular biology, which starts with DNA as a substance which can do two things. It can replicate, which it needs to be able to do to put copies of itself into each new cell as it's being produced. And once in the cell, what it actually does in the cell is it then controls the production of proteins via these molecules called messenger RNA, which we'll come and look at a little bit more closely in a second. Acts as a sort of messenger which takes an imprint of the code on the DNA out into the cytoplasm, where it's used as a template on which proteins are constructed or synthesized on ribosomes. And it's these proteins that carry out many functions in cells and therefore determine that cell's characteristic. Moving on to a next slide, which looks at the whole process of protein synthesis in a little bit more detail, although as you'll notice from the syllabus statement up here, you only need to know an outline of what's going on here, because we come back and study this in a lot more detail in the A2 course. So if I look at this diagram down here, we've got our DNA, which is in our nucleus of our cell here, and what happens is that there are two processes which are involved in making a protein. The first process is called transcription, and that occurs in the nucleus. Okay, and that involves in producing a single-stranded molecule of what we call messenger RNA, or mRNA. That mRNA then diffuses out into the cytoplasm, where it attaches itself to a ribosome, this grey structure here, and on the ribosome, these little black blobs there represent amino acids, are brought one at a time to the mRNA strand, okay, and they are read, the bases here are read or translated into a sequence of amino acids. Remember, there are 20 different types of amino acid, and the sequence of these amino acids determines the secondary and tertiary folding of a protein, and therefore its function. Okay, so the sequence that is laid down in the primary structure of a protein is determined by the sequence of bases on the mRNA. The sequence of these bases on the mRNA is originally determined by the sequence of bases on the DNA. So that forms a little sort of outline of the process of protein synthesis. I address that in a little bit more detail up here. So I said DNA holds coded instructions for the sequence of amino acids in polypeptides. The polypeptides are constructed on ribosomes which occur in the cytoplasm, which immediately gives us a problem because the DNA is in the nucleus, and yet the process of protein synthesis happens in the cytoplasm. The information which is on the DNA is read, or an imprint is made of it by a molecule called mRNA. Okay, so the mRNA is produced using the DNA as a template, and that's known as transcription. And the details of that, how that happens: an enzyme called RNA polymerase binds to the DNA, breaks the hydrogen bonds, unzips the DNA, exposing the DNA bases, and then free RNA nucleotides bind to the DNA by complementary base pairing. Remember, the complementary base pairing is slightly different. A base pairs with U, and T base pairs with A, C with G, and G with C. The RNA unwinds or unbinds from the DNA and diffuses out of nucleus into cytoplasm, where it then binds with the ribosome. And then on the ribosome, the sequence of bases of mRNA is uses as a code to arrange amino acids in a particular sequence, which is the primary protein structure. Amino acids are linked to mRNA by another type of RNA called transfer RNA. So the transfer RNA molecules, which we don't have to know a great deal of detail about, but they're, if you like, they're the sort of adapters which adapt the sequence of RNA bases on the messenger RNA into the sequence of amino acids in the polypeptide, and that whole process is known as translation. So going back to that, we've got the transcription, DNA to messenger RNA in the nucleus, and we've got translation, which is the reading of the messenger RNA messenger molecule by transfer RNA molecules to result in a sequence of amino acids. This diagram here, or this slide here, tries to illustrate that again with other diagrams. So transcription, the idea of transcription is something that happens in the nucleus, which takes DNA and makes a molecule of mRNA, which is an imprint of the DNA code. The messenger RNA then diffuses out where it binds to a ribosome, okay? And on the ribosome, amino acids are converted into a polypeptide in a particular sequence, and that process is known as translation. Again, the whole process is summarized in these diagrams up here. Transcription, this is happening in the nucleus, where messenger RNA has been constructed nucleotide by nucleotide by complementary base pairing between RNA nucleotides and DNA nucleotides, with U base pairing with A to T, C to G, G to C, etc. So this is happening in the nucleus. Out on the ribosome, this is what's happening, is that the sequence of bases on the messenger RNA are being read 3 bases at a time by these special transfer RNA molecules, these blue T-shaped structures there, and these bring amino acids one at a time onto the messenger RNA, which results in a sequence of amino acids, these different coloured squares representing amino acids here. So that concludes our look at DNA.